I'm Steve Williams. I'm going to be moderating this morning's session, and the third moderator in a row comes from the land of the frozen queen. And I think that David Lawrence will have to start practicing his English accent for this afternoon. So thinking about this morning's agenda, how many people here, put your hand up if you know what a homunculus is? So for those of you who don't, a homunculus is a drawing of a human being. And the drawing is not anatomically correct, but it's drawn in proportion to the areas of representation in the brain. So some bits are big, like the hands with lots of nerve endings, and some bits are small. And I think this meeting is actually Larry's homunculus. It shows us the representation of areas in his brain. And the reason I was thinking about this was because I looked at this morning's agenda and I thought, we've got speakers in genetics and nutrition and healthcare reform, and then they're represented in the same part of the homunculus as experts in the art of illusion. I thought, why is that? Why has Larry put those together? And then, of course, you just have to realize that when you walk through the supermarket and you're in the nutritional supplement shelf, the magicians and the illusionists are all there sitting on the shelves promising you don't have to age, you never have to be depressed, your bowels can be perfectly regulated. And as Dr. Shea said yesterday, for genetics, it's the same. We're up to our chests in over-claiming, over-hyping for genetics. And then in healthcare reform, of course, you've got lots of different peddlers of different kinds of illusions. So I think this morning's meeting is about the speakers we have in nutrition and genetics and healthcare are attempting to cut through the illusions. And the speakers who are the experts in illusions are going to tell us how not to get caught. But I think it's all part of Larry's brain. So the first speaker is Tim Harris, and he heads up the the translational medicine group at Biogen Idec. And so please join me in welcoming Tim.
My father once told me about a lecture that he was at when somebody was saying, well, can you hear me? And somebody at the back said, yes, I can hear you perfectly. And the guy at the front said, well, would you like to change places? So we have to be a little careful. Larry also told me that I was keeping people from synagogue this morning. So since I have no idea what that is, I will be as brief and as concise as I can. So no illusions. And I'm not going to stand on these things because I think these are trapdoors. And if I stand on them and I say too much, I should probably disappear. So drug discovery and clinical trials, it's all about genetics. I think it is. So, industry trends. I thought I'd try and set the scene a little bit and give you an overview of how I see the industry trends anyway. And I think I'll stick here. Continuous technology advancement drive new discoveries and clinical applications. That's pretty self-evident. Data-intense integration of molecular and clinical data. Very true. And we're in the realm, as some of you know, no doubt, of what is referred to as big data. That's large, discrete data sets from different kinds of analyses. They need to be integrated and analyzed, and that's causing a lot of concern about how to do that. It's difficult enough analyzing sequence data, let alone putting that sequence data in the context of clinical outcome data. That's the kind of big data problem that's coming. Biomarkers as surrogate markers of drug efficacy or safety. As George Post wrote in I think, Nature Biotechnology, there is a huge amount of crap out there on biomarkers. Most of you are aware of biomarker studies of 10 patients and 10 controls. Some analyte they've measured goes up, and then they make a song and dance about it, publish a paper, and everybody thinks that that biomarker is actually measuring whatever it is they said it measured. Not true. You have to do a lot of validation and replication studies to prove it. The importance of biorepositories and tissue banks, often underappreciated just what some of the rate-determining steps in these kinds of studies are. And I can assure you that if you're thinking about sequencing patients, one of the most rate-determining steps is actually getting them. The sequencing itself is relatively straightforward. The analysis is complicated, but probably the rate-determining step is actually collecting the patients with the homogeneous phenotypes that you want to analyze. And then finally, it's the focus of companies on genetically defined diseases and the coincidence of telecommunications and healthcare. I don't know how many of you have read The Creative Destruction of Medicine book by Eric Topol. It's actually quite interesting. I don't think you'll learn anything, but it's the way he brings the different things together that makes it interesting. So I'm going to give you a vision here, and I think this is a little bit naive, to be honest, but I think it's worthwhile putting it out there for you because it sets the scene for what I'm going to talk about. So the vision is, given that I work for a company that's interested in multiple sclerosis, all multiple sclerosis patients will have their genomes sequenced. All clinical data collected on patients from symptoms, MRI, clinical records, drug use, and effect and outcome will be captured in an integrated database. All biomarker data from each patient, from gene expression to serum analytes, will be in this database, and this will be part of the electronic patient record. And I mean electronic patient record. Outcomes and safety research can be done on that patient record, whether it can be done directly by a company such as Biogen, Idec, or through an intermediary. I think we'll have to wait and see, given all sorts of ethical issues. But nevertheless, we do need to be able to capture that data and help patients as a result of capturing that data. And not only that, but once you've got the data, you'll be able to get a feeling about the cost effectiveness of each medicine as part of that assessment. And one thing that I've been talking to people at Biogen Idec about recently is, with respect to multiple sclerosis patients, it's all very well looking at disability, which is a relatively easy measure to measure, but as some of you may know, cognition is something that MS, or cognitive deficit, is something that MS patients suffer from. And collecting that data and putting it into the healthcare cost is not something that is done routinely. And when you're thinking about pricing our medicines, you do have to have a pretty good idea about what kinds of healthcare costs you're saving by paying the money for the particular medicine with the efficacy that it has. So major drivers for patient stratification then. Financial. The payers will not pay for drugs which do not work in many patients treated. That has been the case in the past. I think it's less so now because everybody is very well aware of that, but that certainly has been the case, and everybody is aware of the situation in Europe with IQWiG and NICE and a variety of other economic controls which make sure that the patients are treated with those drugs that are actually efficacious. Cheap diagnostic tests can save large numbers of dollars of unnecessary treatment. Most of you will be aware of the KRAS mutation and antibodies to the EGF receptor, cetuximab, for example. That was not mandated by the FDA. That was actually done by the physicians who appreciated that those people with KRAS mutations did not respond to the antibody. KRAS wild-type patients did. So there's a vested interest in measuring KRAS wild-type and KRAS mutants to make sure that you only treated those patients in whom the drug would actually work. And given that that drug costs something like $80,000, it's so well worth spending $400 on the diagnostic test. Science and technology, increasing awareness that it's possible to segment patients into disease subset using genetics and other biomarkers for both efficacy and side effects, with all the caveats that I said about making sure that those studies are very robust. Increasing intolerance to side effects in drugs with marginal efficacy in most patients. And I put that there about marginal efficacy because the story I'm going to tell you about Tysabri and PML is very important because Tysabri has extremely high efficacy, and if it didn't, it's unlikely that that drug would still be on the market for reasons that I will go into in a minute. Patient advocacy and interest groups, I believe, and maybe I'm being slightly naive here, that there is a certain amount of physician disintermediation here where patients are able to go to the internet and get a very large amount of information about their disease and are actually in a position to ask the physicians quite detailed questions about what therapy they're going to have and why is the physician going to treat them in a particular way, where they've got information from the internet that tells them how other people have been treated and how well they've done as a result. I think that's coming, and I think that's one of the reasons why we need to be thinking about patient stratification in a rather holistic way. I look at things through a genetic lens as often as I can. There's an increasing interest in the industry in genetically defined diseases, from Mendelian inherited traits all the way through to understanding more about common diseases and the alleles that predispose people to common diseases. Whole genome and exome sequencing is helping to find both common and rare alleles of underlying disease genes. Genome-wide association studies, as some of you know, are being used to find some of the variations in common alleles, but in order to find the maybe genes with a larger effect, which are much rarer in the population, then you have to go to sequencing methods to do that. Both germline and somatic mutations, both new and old, by the way, are being discovered at a rapid rate. And the genetic cause of most diseases with Mendelian inheritance will be found within the next 3 to 5 years. So if you look at OMIM and you look at all the Mendelian inherited traits, it's not going to be long by sequencing those individuals with those diseases that we're going to be able to understand what is causing them, and some of them are caused by novel mutations and some of them are caused by inherited mutations. So there's a slide for you to look at the key technologies, and I really am not going to go through that one by one. It's just really a menu of omics, if you like. And again, I'm going to use Sidney Brenner again because he's a hero of mine. And one thing he said to me, Tim, he said, you know, there's only one omics that's really important, and that's economics. So actually, I've got another one too, because somebody sent me a message the other day and said they were going to buy a new cryotome. And some of you might know what a cryotome is. You make very thin sections of frozen tissue. And I said back in the email, I said, then we can do some cryotomics, which means that you can... Sorry. Anyway, oncology is leading the way. So I'm going to tell you a little bit of stories about how I believe oncology is leading the way, because that's a segue into more complicated diseases such as multiple sclerosis, where you don't have tumor mutations to look at. So most of you are aware of HER2 and Herceptin, and the fact that if you don't have overexpression of HER2, then Herceptin does not work as well as it does when HER2 is overexpressed. You've already heard about breast cancer management using gene expression analysis, either the Oncotype DX test or the MammaPrint test, or indeed a test called PAM50, which is another set of 50 genes which are over or underexpressed, which mark particular types of breast cancer and can be used not only to distinguish different forms of breast cancer, but also help you to understand what is the best treatment for each of those different forms of the disease. There's a test called MERView, which looks at microRNAs and looks at their up or down regulation in metastatic disease, with the idea that you can understand what the primary tumor was if you can only look at the metastatic disease, which is often a problem when patients with cancer come into the clinic. But looking at their MER profile, their microRNA profile, you can get a good idea about where the primary must have been, which is quite important for treatment purposes. You heard about the EGFR mutations and EGFR kinase inhibitors. The EGFR kinase inhibitors work in those patients with those mutations, particularly the Asian female EGFR mutant people were very responsive to gefitinib and other EGFR kinase inhibitors. I've told you about the KRAS study. Measuring mutations in multiple genes to determine best treatment in lung and other cancers is coming. I give you some example of some of the personalized medicine in cancer, if you like. The poster child is BCR-ABL and Gleevec in CML. When that was discovered, I think the prevalence of CML in the US was about 16,000 patients. The incidence was about 5,000 new patients a year. And forgive me if I get this wrong, but I think Novartis were not terribly inclined until Vasila said to do it, to take that drug forward because the market was perceived to be so small. Not true. That drug does something over $3 billion a year and helps a very large number of people, not only with CML, but also with GIST and other diseases where PDGF receptor kinase, which is also inhibited by Gleevec, is overexpressed. EML4 ALK in non-small cell lung cancer. I'll talk a bit more about that in a minute. BCR-A1 and PTEN and the PARP inhibitors. As some of you know, some of the clinical trials that were done on the PARP inhibitors did not stratify patients into those who were BRCA1 or PTEN deletions, and as a result, they got very limited efficacy. Once they went back and looked at the data and saw that it was more active on BCR-A1 mutation tumors and PTEN deletions, then you can imagine what you do next. You do the study in those patients. And the BRAF V600E story with vemurafenib in melanoma, you're all pretty, I'm sure, aware of how important that's been in the treatment of melanoma. So the next step, I think, if you're thinking about oncology and personalizing the medicine, is to anticipate resistance and treat accordingly. And what that means is taking, as we heard from Charles yesterday, serial samples from the tumor to understand when you've moved from a sensitive situation in the tumor to a resistant mutation arising. and there are some genes in which you might expect to see those resistant mutations, and then you can monitor the patient and either have a timeout for the treatment and then go back, or treat the patient with a different agent. So, as you can see from the slide, molecular profiling in lung cancer, this was about 2009, has moved on apace. You can distinguish the tumors pathologically, and then you can distinguish the tumors by looking in their sequence at a variety of different genes and looking for mutations or overexpression in those different genes. This is done now pretty routinely, and as you can see from this slide, it's the integration of molecular profiling into the lung cancer clinic that has, I think, changed the picture as far as lung cancer patients are concerned. Once you've identified mutations, if there are any, then you have a guide to what drugs you might want to treat that patient with. Unfortunately, as you can see from the slide before, there are a large number of patients who still do not have any defined mutations, and they're KRAS wild type, and that makes it very difficult to treat them in anything other than a conventional way. Just a word or two about crizotinib and vemurafenib, because it's important with respect to the diagnostic tests that went with them. If you're going to treat people with crizotinib and vemurafenib, then you treat them if they have a particular mutation in their tumor. In the case of crizotinib, it's either overexpression of ALK kinase or it's the EML4 ALK translocation. You need to be able to measure that, and there are FISH tests and others available for measuring that translocation. And in vemurafenib, since it works only on the V600E BRAF mutation, and then you need to know those patients who have that mutation in their tumor so that you can treat them. It's not actually quite as clear as that. It works in melanoma, but there are colon cancer patients with BRAF V600E mutations as well, and unfortunately, for some reason that's not understood, vemurafenib does not work in those particular patients, even though they have tumors with that mutation, which should be sensitive to the drug. And it probably is. What it tells you is that those tumors have other mutations in other genes that kind of override that, that expression driven by the V600E mutation. So there's a lot of interest in thinking about the oncology picture and thinking about what that means for other diseases. But before I go there, I want to just walk you through some of the new approaches to single gene disorders, because as many of you know, there's been a great deal of encouraging data on those. In particular, cystic fibrosis, where Vertex have developed a drug called Kalydeco, which only works actually on the G551D mutation, which is a gating mutation that prevents chloride ion channel activity, which is what the CFTR does. There are other mutations like G551D, which the drug also works with, but that only accounts for some 7 or 8% of the CF population. It doesn't work on delta 508, which is the most common mutation, which is a deletion in the first nucleotide binding domain. There is, however, a drug being developed by a company called PTC, who filed to go public the other day, called atulurin, which is a nonsense mutation suppressor. It overcomes a nonsense mutation in the middle of the CFTR in another subset of the patients, and it seems to help to encourage that protein to be functional as a chloride ion channel. Watch this space on that particular agent, and as you can see from the bullet point below, it also works on nonsense mutations in the Duchenne muscular dystrophy gene, DMD gene. And again, there's some good data that shows you that dystrophin is expressed from those patients with that mutation when they're treated with the drug. So again, that's looking encouraging, and that goes alongside in DMD those compounds that seem to be affecting splicing, and you can actually splice out some exons with mutations which prevent the protein being formed properly and actually derive a derivative dystrophin which still has residual activity. And companies like Prosensor and Sarepta are pushing those particular compounds forward. They're antisense compounds. In other words, they're compounds that act directly on the messenger RNA. Hemophilia is a recessive X-linked disease, as you know, and talking about the frozen queen, you'll remember that the British royal family, Queen Victoria in particular, had a factor IX mutation, Christmas disease. That's in the blood of the royal families in Europe, just to remind you of that. Biogen Idec is developing long-acting forms of factor VIII and factor IX, and those two BLAs are just in the process of of being filed, and we're very hopeful that those long-acting forms will prove to be useful and helpful for that particular patient population who at the moment have to inject themselves extremely frequently with the less long-acting forms of the factors which are available. Biogen Idec has got another couple of interests in single gene disorders, spinal muscular atrophy for one, and myotonic dystrophy for another, both working with ISIS and both working on antisense approaches of different types. And I think it's fair to say that there is now a third dimension to drug discovery space. It's not just antibodies and small molecule compounds, it's now antisense compounds as well, and siRNA too for that matter, given some of the activities of Alnylam and other companies. So you should think about the drug discovery space as having 3 legs and not just 2. But what can we do for multiple sclerosis? And my sister actually has tertiary MS, and she has actually the progressive form of MS, and there was nothing, she got the disease when she was about 40, there's nothing that can be done for her from a disease-modifying point of view. So I watched her go from a person walking about to a person in a wheelchair with a bladder catheter. That's not why I'm at Biogen Idec, I can tell you, but it certainly helps to think how lucky we are to be able to walk around when she can't. So we have things to do for this disease, and the thinking is, what can we do for multiple sclerosis using the kind of thinking that I've been talking about? And there are a couple of things that we can think about. First of all, looking at common susceptibility alleles and how they guide you as far as disease stratification is concerned, and then looking for rare familial genes for progressive forms of the disease. If you like, you can consider PPMS to be an extreme phenotype of MS, rather like optical neuritis, which is another form of the disease, which is sort of an extreme phenotype. And as you know, from a genetics point of view, looking at extreme phenotypes can often help you to understand the pathology of the more common forms of the disease. So it'll help us to find better points of intervention, and it should give us an opportunity to stratify patients based on their genetic background. Biogen Idec is going to be in a pretty privileged position fairly soon, because we're going to have 5 or 6 different agents that can be used to treat multiple sclerosis of different kinds, relapsing remitting MS in particular. And you have to wonder how you're going to stratify the patients that are going to be treated with our drugs. Right drug, right time, right patient. It's actually pretty, since you've got both small molecule compounds like Tecfidera, which is dimethyl fumarate as an agent, you've got Tysabri, which is an antibody, and you've got Avonex, which is beta interferon. Different types of agents which you might imagine would do different things with different patients. At the moment, it's not really clear when we can identify which patients will do better on which particular drug, except to say that Tysabri is probably the most efficacious drug out there and tends to be used after Avonex has been used and the patient has relapsed or is not continuing to do well on that agent. But anyway, you can understand the problem that we have. It's an opportunity, but it's also a problem. So what we have to do is to try to find out more about the patients on our drugs so that we can stratify them into those that we think will do well on one drug and maybe not so well on another. And I think there's a commitment to companion diagnostics to help us to do that, and I'll give you an example of that in a minute. And I think there's payer pressure to find stratification markers because they do want to reimburse those drugs that work, and they certainly are not interested in reimbursing drugs that do not work particularly well. So let me tell you a little bit about Tysabri. Some of you will be very familiar with this, I'm sure. It's a monoclonal antibody against VLA4. It binds to VLA4 on the surface of lymphocytes, and it blocks interaction with VCAM, and those white blood cells don't extravasate into the bloodstream. So they don't get into the brain, where those ones that are reacting with myelin basic protein or other parts of myelin can't then stimulate demyelination, causing lesions and then MS. So it prevents that happening. Some patients on toluzumab, which is the trade name, the non-trade name for Tysabri, develop a disease called PML, progressive multifocal leukoencephalopathy, which is not good news and is not always fatal, but always leaves you with some kind of neurological sequelae. So you want to avoid it. And it's caused by a virus called JC virus, which is a polyoma virus, and it's a rare event. About 1 in 1,000 patients are likely to get PML as a result of being treated with Tysabri. It's related to how long they've been on the drug. It's related to prior immunosuppression. And it is related, it turns out, to having antibodies to JC virus. So what Biogen Idec has done is to develop a test that looks for antibodies to JCV. And if you are a patient who has been on Tysabri for 3 years, prior immunosuppression, and you have antibodies to JC virus, then it is contraindicated and the physicians will probably not prescribe Tysabri. There is some ongoing work to look at levels of antibody. There seems to be some correlation of levels of antibody with risk of PML, and we're in the process of trying to work out just what that means. But that's a good example of a diagnostic that goes along with a highly efficacious drug. And as I said in the beginning, if Tysabri was not as good as it is in controlling disease, I think it's unlikely that it would have got back on the market, which it did when it was withdrawn owing to the first signs of PML that were seen in the early 2000s. So it's a very good story, meaning it's important for the patients. That's what I mean by a good story. But it demonstrates the need to be very diligent about looking for markers that can help you to keep efficacious drugs on the market by finding markers that mark those people who are likely to suffer from a serious side effect. PML does occur in other diseases as well, where immunosuppression occurs, either drug-induced or caused by infectious agents such as HIV. Much less common in HIV now, because, especially in the US, because they're treated with triple therapy and they are not as immunosuppressed as they were at the beginning. So there are a number of themes that we're following for risk stratification for PML. Suffice it to say, one of them is with SomaLogic, looking for somomers that can give us a profile that marks those patients who are more likely to suffer from PML than others when they're on Tysabri. We're in the middle of doing those experiments. We're also sequencing those patients who have got PML as a result of being treated with Tysabri to see if we can see any genes that seem to be more common, or mutations in genes that are more common in the patients who get the disease than those patients who do not. That is not that easy because there aren't very many patient samples to look at. So it invokes all sorts of things about just how many genes there might be and frequencies, etc. But suffice it to say, and I'm going to skip over the slide, we've done about 30 full genomes. A group in Oxford have done about 30 full exomes of such patients, and we have just about to join the two data sets together. But as far as I understand it, neither we nor Oxford have actually discovered any particular gene that seems to be a culprit for marking those individuals who are going to get PML as a result of being treated with Tysabri. So we're not there yet, but that doesn't mean to say that that approach won't work. I'll just talk a little bit about common alleles. As some of you know, very large genome-wide association studies have been done in a lot of autoimmune diseases, multiple sclerosis as well, and a number of different SNPs have been found in something greater than 50 different genes. Now, if you look at those SNPs and you assume, well, they're not actually in the genes, they're close to the genes. If you assume that those SNPs mark those genes, and that's an assumption, but a reasonable one for now, then you can put those genes into different pathways, as has been shown on this slide, and then you can ask yourself the question, does having slight variation in the expression, for example, of a gene that is marked by a susceptibility marker make some sense in predisposing an individual to a disease like MS? And unfortunately, as is the case with most candidate gene studies, you're always one gene away from a sensible hypothesis. But having said that, you can look at the slide and you can take it for what it's worth. Look at all the interleukins, look at all the different gene pathways that are involved. And what we did was to overlay some of the drugs that we've got, we're either developing or have been developed, and tried to make some sense of the susceptibility alleles with respect to the disease. What has not been done and what needs to be done is very large genome-wide association studies where you not only look at multiple sclerosis, but you also look at outcome with treatment with a particular drug to see whether you can see any alleles that actually mark good outcome. They might actually have a much larger effect than these genes do and might be much easier to find. But no one has actually done those studies yet. We're in the process of setting up to do them. So at the moment, the common alleles don't help you a whole lot in putting patients into different categories. Having said that, it will be very interesting to look at patients who happen to have the wrong allele at every single one of these susceptibility sites and ask the question, what is their risk and the sibling risk that goes alongside that if they're in a family, and look at how they might respond to particular drugs compared to people who don't have all the susceptibility alleles. Because you don't have to have the susceptibility alleles to get MS. They're just susceptibility alleles. You can still get the disease without them. Same applies to the HLA, which is the most highly associated locus in the human genome for autoimmune diseases in general. I will refer you to a couple of papers in Nature recently on the effect of salt on the expression of some genes in MS. Very exciting study which just goes to show you that the environmental influence on the disease is very substantial. Both sunlight, vitamin D, salt seem to be very important in the development of the disease. So take a look at that if you're interested in environmental genetic risk of those kinds of disorders. Will genetics allow the segmentation of other diseases such as amyotrophic lateral sclerosis? Some of you know that this disease has been analyzed genetically in some detail. There are familial genes that have been found. There are genes involved in the spontaneous diseases you can see from the slide, and some of them are really becoming quite interesting. There's a drug called C9orf72, which, as its name suggests, is a drug that has a gene that has no function. It just happens to have an ORF name. It turns out that that gene is much more common in the population than you might expect, and there are even siblings of ALS patients who have the same C9orf72 mutation who don't have ALS, or at least they don't have ALS yet. So the penetrance of some of these mutations is rather low, and that makes the whole thing somewhat more complicated. But what we're doing, as you can see from the slide, is we're sequencing something like 1,000 patients, full exome sequencing, to look for other mutations that may be involved in the pathology of the disease. And the purpose of that is not necessarily to allow us to segment the patients into different buckets, but actually to see whether we can get some kind of concentrated view of the pathology of the disease, because you're basically walking into a room called ALS by going through doors with a particular genetic marker on each of the door, SOD, C9orf72, TDP43. It doesn't matter what the gene is, you've got to walk through that door in order to understand the biology, and it's the way those pathways converge that I think is going to help us to discover new drugs to treat the disease in general, or we're going to have to think about treating each individual type of ALS, like, for example, developing a drug that affects the C9orf72 gene, which seems to be involved in RNA processing or RNA binding. Might also be that expanded repeat might also be translated. There's some evidence that it's translated, which makes it even more complicated. So you've got gain of function, loss of function that are being invoked for that gene. There's a lot of work to do, but we may end up getting a C9orf72 directed medicine. We might use an antisense approach to that as well, as ISIS and others are doing. So I'm going to finish off by talking a little bit about Alzheimer's disease, because this is a bit of thinking about how you can use genetic information to help you with setting up a clinical trial. So as you know, there are early onset familial forms of the disease caused by defects in certain genes, PSEN1, PSEN2, APP, tau, and interestingly enough, as we heard yesterday, Down syndrome too. One of the sequelae of Downs when they get into their 40s and 50s is that they get a form of Alzheimer's disease. They also suffer from other neurological diseases, which is why I asked the question about alpha-synuclein, which is involved in Parkinson's disease, is on chromosome 21 and is triplicated in not all Downs patients, but some Downs patients. That's why I asked the question yesterday. There are also some susceptibility alleles, ApoE4, and a new gene called TREM2, which has been found very recently in the AD population. And those susceptibility alleles are fairly high-powered alleles in the sense that they provide a lot of susceptibility influence, 10 to 30-fold increase in relative risk for homozygous ApoE4 patients. If you get to 80 and you're an ApoE4 homozygote, you have a very high chance of getting Alzheimer's disease. It's not quite a disease gene, but it almost is. Some of you know the amyloid hypothesis. I'm not going to walk you through it, but it's clear, and actually the genetics never lies. The genetics tells you that it's true, that the deposition of amyloid is an important part of the disease. The question is, what comes first? Is it the deposition itself that is causing the disease, or is it something before amyloid gets deposited that is causing the disease? And so it's useful to look at this slide about the progression of AD. Looking at some of the familial forms of disease, you can certainly start to understand that the disease occurs very early. You can't detect it, but there's a lot of, if you like, pre-disease activity going on well before the mild cognitive impairment occurs, and you start to get amyloid deposition and then frank dementia in the patients. So, as you know, some of the recent failures of some of the drugs that have been tried, the antibody drugs in particular, are not only due to poor clinical trial design, but they're also due to the fact that people perhaps went into the patient population too late, when you really can't affect the pathology too much by binding to beta amyloid because the damage has already been done. So the amyloid hypothesis is alive and well. It's knowing when and where to intervene that is the question. And I stress that point because a lot of people have said, and you've read the editorials, oh, the antibodies failed, therefore the amyloid hypothesis is wrong. The amyloid hypothesis is not wrong. The genetics tells you that the amyloid hypothesis is absolutely right. What it doesn't tell you is how do you get after the particular hypothesis? How do you intervene? It doesn't tell you that. The antibody trials say more about the antibodies and trial design than the hypothesis. We need rapid proof of mechanism. Clearing different forms of amyloid is possible, and you can now use imaging techniques to look at the presence of amyloid. You'll be interested to know that about 30% of the patients in those two antibody trials did not have any plaque anyway. And they might not even have had Alzheimer's disease, actually, which brings up questions about how do you do clinical trials? Do you really rely on CROs for those kinds of trials, or do you think about them as experimental medicine trials? They're clinical experiments. They're not clinical trials, where you actually need to have some control over what goes on in the centers that are doing those clinical trials. So we need rapid proof of concept. We need some sophistication of phenotyping required, and we certainly need a set of entry criteria which make you convinced that the patient actually has the disease. Mild cognitive impairment, you need to see the plaque, you need to measure p-tau and A-beta 142 in the CSF, and you need a cognitive impairment test that is relatively easy to do and correlates with dementia as far as outcome is concerned. There are multiple approaches to APP modulation. I've talked about immunotherapy. There are also enzyme inhibitors involved in the process of inhibiting the process of amyloid, beta amyloid is formed as a precursor. These enzymes are involved in cutting that precursor up into different forms. And the enzymes that inhibit that enzyme, the beta enzyme, inhibitors are being developed by Eisai, Merck, and Lilly. And on the face of it, looking at the data that they've got on amyloid, they certainly seem to reduce the amount of amyloid that is deposited. Whether that translates into any effect on Alzheimer's disease and dementia remains to be seen. But they're looking encouraging, it has to be said. And I think the view is that you may have to use both a BACE inhibitor and immunotherapy in treating AD. There are a number of different prevention trials going on using genetics. I'll leave that for you to look at. Using those familial mutations that I talked about in the middle, you can actually collect patients who've got particular familial mutations, and then you can do clinical trials of your agents in those subsetted populations. Your marketing group will tell you that that's not going to help you very much because only a very small proportion of the Alzheimer's disease population actually have these mutations, so there's no market for a drug. What I will tell them, and I do, is that actually we want to know whether this agent is actually going to work in the general population. And if it doesn't work in the subsetted population, the segmented population that have the mutations, you can bet your life it's not going to work in the general population. So don't waste your money, and do allow me to do my small trial in this genetically defined population, because I have to do that to gate the outcome in order for me to be interested in spending the sort of money I'm going to have to spend to demonstrate activity in the general disease. So that's basically how I believe we should use genetics in clinical trials. It not only applies to Alzheimer's disease, it applies to other diseases as well, and we've all got to get more comfortable with thinking about taking those subsets, getting proof of efficacy, proof of concept, and then moving forward into the bigger population. So at that point, I'll stop and thank the Translational Medicine team, our Genetic Scientific Advisory Board, who you can see up there, and an enlightened executive team at Biogen Idec who believe in translational medicine and what we do. And I thank you for listening, and I thank the company for allowing me out to speak, because I often say things that they probably don't want me to. I didn't today.
It's okay. I was struck by, I never thought about this before, but I was struck by the difference between a clinical trial and, as you phrased it, a clinical experiment. It's an incredible idea, and you couldn't help but think about how hard it is for you guys, as good as you are, or seem to be, to get all the best information in spite of your SAB for clinical development and your translational SAB. And I was thinking about Matt Todd's talk yesterday. And so here's now finally a question. Has anybody in a big pharma company ever thrown out into his kind of space for open discourse, not even just with the disease advocacy group, but the world, and said, we're about to do, what do you think? Has anybody ever tried that?
Well, they're beginning to. I think there's a number of companies that are beginning to do that. As you know, there's a group called 23andMe who are involved in looking at genotypes with respect to particular traits like does your urine smell when you eat asparagus, which is not terribly helpful for drug development, but they have actually, as you know, for reasons, historical reasons with the company, are interested in Parkinson's disease. So we've actually, I think Sergey Brin's family has an LRK mutation which predisposes individuals to Parkinson's disease. So they have an interest in PD. We have actually worked with them to see how easy it would be. It's a logistical study to see how easy it would be to use the 23andMe database as a way of recognizing Parkinson's patients and then identifying them, bringing them into a clinical study as a result of knowing what their mutations are. So you could get alpha-syn overexpressors, you could get LERT mutants into a place where a small number of people may give you enough people to give you a decent clinical experiment where you don't just look for efficacy, you actually measure things as well in the patients that you bring in. I think that's a very interesting concept. I help to push that forward. I think we can do that much more with the groups, the advocacy groups that have patients that they know. I think patients are the least worried about entertaining that idea. It's the lawyers that don't like it much because of the anonymity and all the different things that are a problem or perceived to be a problem. You speak to the patients, they couldn't be more delighted to take part in a study like that, get to the clinic, get the whatever clinical phenotype workup they need, and then treat it or not with the drug that's available. I think it's coming, Larry. I think virtual clinical trials, as it were, is something for the future. And if we like to think about it, let's do virtual clinical experiments first, and then do virtual clinical trials later.
So I wonder if you could comment on the fact that, say, 5-8 years ago, most companies, including probably Biogen, were not interested in rare genetic diseases, and now, you know, a Vertex drug that affects or that treats a very small fraction of cystic fibrosis population, which by yesterday's standards, it's a very small disease to begin with. And the same is true, for, say, for Sarepta, where I'm from, where only, and then the next cohorts of the patients that could be treated probably will be even smaller, and yet it seems that that's where the whole field is going to.
Do you want the cynical answer or the not cynical answer?
Both.
No, I don't think I'll give you the cynical answer. I think I'll give you the non-cynical answer. I think it depends on the efficacy of the treatment. I understand it that Kaleido works really well in the G551D mutation patients. If you had a offspring who happened to have that mutation, you would be more than happy to treat them with the drug. Your expectation would be that your healthcare provider would reimburse the $330,000 that it costs per year to treat your offspring. I think that's quite reasonable. I think that invokes a particular argument about the cost of the healthcare costs in this country and what our expectations are. Maybe this is not the time to go into that philosophical argument, but it does beg that question. And I think that provided you can demonstrate very high efficacy in the subsets that you're treating, then it's perfectly respectable to do that. Where it becomes less respectable is where the effect is really marginal and you're still expecting the patient to pay, or somebody to pay, $330,000 for the drug. And I can tell you, I was quite disturbed by that number when I first heard it, but then I was less disturbed as I heard that actually it works really well in those patients who are taking it, which abrogates that problem as far as I'm concerned, provided someone will pay. And that is the question. Who's going to pay?
My comment was that if I understand that the patient, of course, is ready to do whatever it takes to have their offspring treated, but you as a company which needs to, you need to invest in $100 million in a clinical trial without knowing whether your treatment is effective, right?
You do that anyway.
That's
I mean,
high risk.
for most diseases you're treating, you don't really know whether it's going to work or not.
Right.
I think you've got a higher expectation if you're working with a drug that has been tailored to a particular genetically segmented population. You've got a higher expectation of it working. So I think that justifies the experiment, give me the clinical trial. The question of how you price the entity once you've demonstrated efficacy is a whole nother question.
Final brief question, please.
The question has to do with your comment about MS. And so we know for most autoimmune diseases, we know a lot about the middle and the end of the disease. We know that Tysabri will inhibit VLA4 binding to VCAM1. But the question is, why are there activated T cells that can respond to myelin sheath or other relevant antigens in the circulation in the brain in the first place? And the question is, do you believe that, to use your metaphor, that walking through or opening all of those doors on gene expression can give us an answer to that question?
Well, you specifically said gene expression, and I don't personally believe that walking through the door marked gene expression is necessarily going to help you in of itself. The way I like to think about gene expression data is you have to look at it in the context of other data that you can provide. For example, genome-wide association study data. You need to look at transcriptional array data in the context of SNP data, in the context of proteomic data. I don't think that proteins are the be-all and end-all by any means, but I do think you need to be appreciative of the changes that occur in as many different molecular entities as you can measure. miRNA, transcriptional arrays, somomers to measure different levels of different proteins in different fluids. All of those things will help you to understand. That's what I meant by, if you like, big data in the research environment means getting each of those data sets from a number of well-controlled studies and interrogating the data as a whole. And some of these crowdsourcing methods that are being talked about, provided you give the crowdsourcing individuals very high-quality data, then they might be able to demonstrate some associations that you have never seen before. But the problem, as you know, is crap in, crap out, and missing data. In most of these experiments, there's too much missing data, and your power evaporates in a heartbeat.
Thanks, Tim.